Well, hello everyone. It's really great to be at your homes talking about science with you today. I'm going to talk about the integrated stress response in cardiometabolic syndrome, and you may be wondering why you're seeing all these organelles, because this is really an integrated stress response of the organelles that I'm going to talk about. So a little bit of introduction to my lab. So we have a unique situation where the studies started in Bilkent University in Ankara, Turkey. That was my first lab when I was an assistant professor. And we published some really nice work from there. And then opportunity brought us to Cedars-Sinai Medical Center and UCLA in Los Angeles, California. And we published our most recent work on intercepting the lipid-induced integrated stress response from there. So I'm going to talk about this work, and so the people I want to highlight and thank are in these slides. So Inji was a graduate student who led this work. It was her PhD thesis, so I really wanted to point that out. And also Allele is a current PhD student who contributed a lot to the mechanisms that I'm going to talk about. So many thanks to these wonderful students who are now going on to their postdoctoral positions. So my lab is interested in cardiometabolic syndrome, which is a modern challenge for the modern man. Because of obesity, we see that there's a cluster of diseases that the modern man and woman has to deal with. So obesity brings with it a cluster of diseases such as insulin resistance, fatty liver, hypertension, and atherosclerosis, which I work on. So a lot of the mechanisms that I'm going to talk about connects these cluster of diseases in one way. From obesity to atherosclerosis is where I'm going to focus, but you can make the connections as we go on. So when there is an excess of lipids or excess of calories, the dilemma is for many cells is to store or not to store it. There is one cell type called adipocytes, as you know, which has incredible ability to take up the excess of calories and store them as lipids. But other cells, when they are experiencing a flux of lipids in the circulation, don't have this luxury. So lipids build up. They can't be properly stored in the pancreas beta cells. They can't be properly stored in the muscle or the vascular endothelial cells or macrophages. So we see a lot of toxicity that comes from not being able to deal with the excessive flux of lipids, which we call lipotoxicity. So lipotoxicity has a consequence, and obesity has a consequence. So we have seen over the last couple of decades a lot of publications that demonstrate that there is a metabolically induced chronic and low-grade inflammation that is causally associated with this cluster of diseases around obesity, including diabetes and atherosclerosis. So the interesting thing about this metabolic inflammation or metainflammation is that it involves not only metabolic cells like adipocytes, not only immune cells such as macrophages, but we see other cells like metabolically active cells like adipocytes and liver cells, which become places where you can see secretion of cytokines and chemokines. So it's a very unusual form of inflammation that associates with this disease cluster. So my lab really focuses on how the immune cells are activated by metabolically challenging conditions like excessive lipids. And these macrophages have been shown to accumulate lipids and show signs of lipotoxicity and inflammation in obesity, in the heart, in the vascular vessel cells, such as macrophages infiltrate the atherosclerotic plaques, or in the ischemic heart. But they're also seen in the obese brain as glial cells that are activated. Another thing that's very interesting is that whenever we see inflammation, we often see it being intertwined with signs of organelle stress. So when you look back at the studies over the last couple of decades, you will see that it has been shown that both inflammatory activation and signs of organelle stress, such as ER stress and mitochondrial stress, have been both associated with tissue dysfunction in metabolic disease, but they have also been shown to be causally responsible for the metabolic dysfunction and inflammation in these organs. So for example, we know that ER and mitochondrial stress can lead to beta cell dysfunction and beta cell apoptosis. And my own studies contributed to the field by showing that ER stress contributes to atherosclerosis progression. So how do we envision organelle stress leads to metabolic and inflammatory dysfunction? So if this is a really nice review that I always go back to. It was written by one of my lab mates, Sunang Fu, in 2012. And Sunang envisioned metabolism as a metabolic roundabout, where insulin can really promote this roundabout of metabolic intermediates to come in and go out and be exchanged for other molecules. And as we know, cytokines can inhibit this metabolic roundabout. And this metabolic roundabout happens both in the cytoplasm, but also compartmentalized in organelles like ER and mitochondria. So you can imagine that stress in these organelles would really block this roundabout. So this is how things are very, very interconnected with organelle stress. Well, the dysfunction of a single organelle can lead to the dysfunction of other organelles. We used to think when I was a medical student years and years ago that organelles are somewhat autonomous, disconnected compartments, but this is not true. As we know from studies, and I want to show here, organelles in real time, you can see, are very, very connected with each other, and these connections are super dynamic. So the dysfunction of ER can easily lead to mitochondrial dysfunction, and mitochondrial dysfunction can easily lead to ER dysfunction. And the reason I want to point out ER and mitochondria is that you can see here, ER and mitochondria are found in close proximity to each other in the cell. They come very close, but they don't really connect physically. They make synapses almost. And this is a really, really important interface for inflammation or immunometabolism, and obviously for cardiometabolic diseases. Because you can see here, mitochondria on the top and ER at the bottom. This interface is where metabolism happens between these organelles. So they collaborate on lipid synthesis, such as cardiolipin and other phospholipids. They exchange metabolites, they exchange calcium to communicate, they exchange ROS and ATP for important communication reactions. And this is also the interface where you can surprisingly see some innate immune regulators, like the MOBs, which sits and regulates antiviral innate immune response on mitochondria, as well as the NLRP3 inflammasome complex, which sits at the MOMs, not in the ER as you see here, but actually on the membranes at the cytoplasmic space. This is a perfect location for these innate immune regulators to sense what is going on in these organelles and coordinate metabolism together with an inflammatory and immune response. So organelles form really important immunometabolic interfaces, such as ER and mitochondria. And now that we are talking a little bit about the inflammasome, which sits on this ER and mitochondrial interface, it has been, it's very well appreciated now that activation of NLRP3 inflammasome and the cytokines that are secreted as a result, like IL-1 beta, can lead to increased lesion formation in atherosclerosis and have been associated with plaque rupture. So this brings me to a stress response which impinges on the organelles and the activation of the inflammasome. So today I want to talk about the integrated stress response, which is not visited in cardiometabolic disease a lot, but it should be. So this response is called integrated because it really integrates a lot of signals at the level of phosphorylation of the eukaryotic initiation factor 2-alpha, which you can see in the middle. So what kind of signals are received? These are stress signals such as the deprivation of amino acids, a viral infection, deprivation of heme in the cells, or stress in the endoplasmic reticulum. So the second level is sensing of these signals by ISR regulators, and there are 4 kinases which senses each of these signals. These are called eIF2-alpha kinases. I'm going to talk about one kinase today called PERK kinase, which senses the stress that happens in the endoplasmic reticulum. As a consequence of activation of these ISR regulators, we see that they phosphorylate the eukaryotic initiation factor 2-alpha. This is a really important phosphorylation, and how do we know this? It is highly regulated. As soon as the phosphate is on, there are phosphatases that remove it. And this highly regulated phosphorylation controls cap-dependent mRNA translation. So this is the core of the integrated stress response. When there is stress, phosphorylation of eIF2-alpha stops cap-dependent mRNA translation, but through very unique regulation, some mRNAs, such as the ATF4 transcription factor, can be translated. And these are the executors and effectors of the integrated stress response. ATF4 is very central transcription factor which regulates chaperone production, antioxidant response, and now we're learning that it regulates a lot of things in the mitochondria. So this is the stress response that I want to talk about. It's called integrated stress response because it integrates a lot of stresses, but it also integrates ER and mitochondria in a deep mechanistic way. So the arm of the integrated stress response that I'm going to talk about is regulated by PERK kinase, which sits on the membranes of the endoplasmic reticulum. So this is also part of the ER's unfolded protein response. In this way, PERK kinase can communicate problems in the ER regarding folding and others to the translational apparatus to generate chaperones and antioxidant response. So a little bit more about the integrated stress response. As I mentioned, eIF2-alpha is at the core of this integrated stress response, and we just viewed it in a linear fashion from the kinases to the eIF2-alpha. But I just told you that it also connects this phosphorylation back to the unfolded protein response in the ER. In the recent years, when I say recent, in the last 3 years or so, we have been learning a lot more about how this phosphorylation connects all these responses to the mitochondria. One of the well-known connections comes from eIF2-alpha to ATF4 to ATF5, which regulates the mitochondrial unfolded protein response. Probably this is not surprising because when you expect unfolding protein response and proteostasis happening in the ER, it would be coordinated with protein production proteases in the mitochondria. Another really interesting thing we learned in the last year is that this phosphorylation also drives mitochondrial supercomplex formation, meaning more productive mitochondria in terms of energetics. Today, I'm going to talk to you about our findings that this response can limit mitochondrial clearance, which is mitophagy. And I'm going to end my talk with our current findings that lead this response to communicate to the nucleus by way of mitochondria, but this is ongoing studies, so I'm not going to have a lot of time to speak about it, but I'll introduce these to you. So the study that I'm going to talk about used a lot of technologies that were available to us at the time to really dissect the integrated stress response in macrophages as they were challenged by lipids and hyperlipidemia. And our biggest question was to figure out if this response plays a role in atherosclerosis progression. So I'm going to introduce you to tools that we used throughout the study. So we were really in a lucky decade because of the interest in the signaling, especially from the cancer field, has led to the discovery of a lot of small molecule regulators. So PERK kinase has several small molecule inhibitors, one of which we will describe and use in the study. And because of specificity issues with inhibitors, we also developed in my lab an ATP analog sensitized PERK kinase allele mouse model. So you probably don't hear this a lot because this is the only example of this in an in vivo setting. There have been studies with this technology, which is known as kinase switch. But a lot of those studies are going to be in the cells. And this was the first time we used this in vivo in a chronic disease model. So we generated a mutation, a double point mutation in the PERK kinase cavity. So normally kinases have ATP binding domains. And this domain, because it binds ATP, is highly conserved in the 500 plus human genome. So if you can predict how ATP interacts in this cavity in one kinase, you can predict how it's going to interact in the other cavity of another kinase. And when you make a point mutation, you can actually introduce a bulky side chain ATP molecule, which will still fit in the cavity. So in the wild type, you can see that a bulky ATP analog does not really have a good fit. But when we make this mutation, point mutation, in a gatekeeper amino acid residue, we can fit in a bulky ATP analog. Therefore, we call this an ATP analog sensitized PERK kinase allele, which we built in as a transgenic mouse model. This will allow us to use, this will allow us to use PERK kinase mice as a kinase switch where we add the bulky ATP analogs that can't be used to phosphorylate substrates as inhibitors. So this on and off model combined with a genetic mutation gives us specificity that might be lacking with small molecule inhibitors. So those were approaches we used to modulate the sensors or the very upstream of the integrated stress response. But we also targeted eIF2 alpha, which is the core of the integrated stress response. And to target eIF2 alpha phosphorylation, we collaborated with several labs which had amazing new small molecules. So we used Sephin-1 that was developed by Anne Bertolotti, which targets eIF2 alpha phosphorylation by targeting phosphatases. And we use ISRIB, which targets the signaling below eIF2 alpha. So I'll tell you how these works. So Sephin-1, which was discovered by Anne Bertolotti's lab, inhibits a phosphatase that is induced in ER stress or stress responses. This phosphatase, called GAP34, is inhibited by Sephin-1. So when that happens, the phosphorylation of eIF2 alpha under stress condition remains. So the sustained phosphorylation allows us to see a gain of function in this pathway. And if you inhibit the pathway with ISRIB, what this does is ISRIB is actually an activator of a GEF, which targets eIF2 alpha, GTP-GDP exchange. So when you activate the GEF, you do not really change the phosphorylation on eIF2 alpha or the upstream kinases. So the signaling upstream is intact, but you block the effector molecule production, the ATF4 translation and translation inhibition of cap-dependent proteins. So these tools allow us to dissect things great in a way from cells to animals. And I also want to highlight ISRIB as a molecule, because this is probably one of the most beautiful examples of a small molecule discovery where we know so much about the structure of the molecule and how it interacts with its target, which allows us to carry these studies to the next step. So here you can see 2 molecules of EIF2B, which is a GEF, in beautiful mirror image positioning to each other. So these 2 molecules of EIF2B are coming together because the small molecule ISRIB acts as a molecular staple. You can see it right in the middle. So this allows us to understand a lot about this small molecule that we're going to use in our studies. So with these tools in hand, and I had 2 very intelligent and motivated PhD students in Ankara, Turkey, who have now moved on to postdoc positions in New York University and Bilkent University. So Inci and Özlem, they tried to test this technology to see if in macrophages, if you inhibit integrated stress response, can you prevent inflammasome activation? So you can see here, wild-type PERK and the mutated form of PERK can be activated by fatty acids. When macrophages are exposed to fatty acids, excessive fatty acids, saturated forms, you can see this response is activated. We knew this from many studies before we started. And if you inhibit the mutated PERK with the bulky ATP analog, you can see here, NAPP1. You can inhibit it, but you don't inhibit the wild type. These are macrophages from these mice. You can clearly see that leads to inhibition of IL-1 beta, mature production, and caspase-1 active 4. So this told us that our model can really work in macrophages, and we could see that PERK inhibition by this approach has a consequence on inflammasome activation by lipids. So we validated this in many, many ways, and I'm just going to show you a few validations. You can see here that if we use a knockdown approach in primary bone marrow-derived macrophages, PERK inhibition also blocks a lot of the caspase-1 production. And also, if we inhibit the ISR core with ISRIB, you can clearly see that IL-1 beta production is dramatically reduced, also with ELISA. And you can also see this if we target the very downstream of the ISR, an effector molecule ATF4, that inflammasome activation is blocked, as well as IL-1 beta production. So these studies validate that our transgenic macrophages were working fine, and indeed PERK was important for lipid-induced inflammasome activation. Okay, so this is exactly what we found out, that if we inhibit PERK or ATF4, we could block NLRP3 inflammasome activation. So the next question we wanted to ask was, what is the consequence of this? Now that we have this mouse model, can we see the consequence of this on atherosclerosis? So we took these transgenic mice, we put them on Western diet for many weeks, from 8 weeks of age to 22 weeks of age. And towards the end of our diet, we gave them the bulky ATP analog by intraperitoneal injections. You can see here that this was very effective in blocking EIF2 alpha phosphorylation, which was quantified here. And this had a serious consequence on reducing lesion size in the in-phase aortas, as you can see here. So switching off PERK kinase using this technology showed us that we can reduce atherosclerosis progression. So now we used the available PERK small molecule inhibitors to validate our observations. And you can see here that if we treat the mice on Western diet with the PERK inhibitor, this greatly reduces atherosclerosis lesion area in the end-phase aortas, and also the total plaque area in the aortic root sections you can see here, which is quantified. If we go down to the core of the integrated stress response and inhibit the stress response with ISRIB, at several dose studies, we found that it could also reduce lesion size. So wherever we target this pathway, from upstream to the core to the downstream, we can prevent atherosclerosis progression in mouse models. And these studies show that there was a lot less foamy macrophages infiltrating these plaques. And together with this, we saw a great reduction in IL-1 beta in the plaques and IL-18 in the secretion, in the circulation, sorry. So far I've told you that from macrophages to the atherosclerotic plaques, inhibiting PERK or ATF4 by multiple methods, including a kinase switch transgenic mouse model, small molecule inhibitors, small molecule activators, we could see that atherosclerosis and NLRP inflammasome was greatly reduced. But I didn't tell you how this happens. So now I want to talk about how this happens. And the first thing we did was, together with Allele and Inji, examine what was known about inflammasome activation. And NLRP3 inflammasome is known to be very sensitive to stress in the mitochondria. So studies have shown that mitochondrial stress leads to mitochondrial DNA release, mitochondrial ROS release, all of which have been shown to activate the NLRP3 inflammasome, which sits at the ER and mitochondria membranes. Interestingly, if you induce mitophagy, which leads to the clearance of the stress mitochondria, this prevents NLRP3 inflammasome activation and the secretion of IL-1 beta and IL-18. So mitophagy is part of the normal life cycle of a mitochondria. When mitochondria is damaged, one of the ways to deal with the damaged mitochondria is to engulf it in a phagophore and turn it into an autophagosome and clear it with the help of lysosomes. This process is known as mitophagy. One of the clues that we had that this might be a process involved is that when it's been known for a long time that treatment of cells, macrophages with saturated fatty acids like palmitate leads to mitochondrial ROS production. And the clue was that if we inhibit PERK, we could see this greatly diminish. So mitochondria was releasing a lot less ROS if the integrated stress response was blocked. When we looked at the cells, these macrophages that were treated with palmitate and inhibited with a PERK kinase inhibitor, we could see really things happening to the mitochondria very clearly. What we saw was that inhibition of PERK led to a lot of mitochondria to be engulfed by the lysosomes, suggesting there was an induction of mitochondrial clearance in these cells. But does that have any consequence on mitochondrial ROS production? To test this, we use Parkin knockout macrophages and wild-type macrophages. Parkin is one of the important initiators of mitochondrial clearance. And so if you have a knockout, you don't see mitophagy happening through this general mitophagy pathway. So we can clearly see here that mitochondrial ROS reduction with palmitate happens in the wild-type cells, and it also happens in the Parkin knockout cells. But when we inhibit PERK, wild-type cells reduce their mitochondrial ROS production. But in the Parkin knockouts, this is not reduced. So this clue suggested to us that mitophagy was an important way for PERK to help clear damaged mitochondria by the fatty acids and to prevent the mitochondrial ROS leakage. And did that have any consequences to inflammasome activation? So we took the same cells, wild-type macrophages and PERK and knockout macrophages. You can clearly see here that palmitate induces IL-1 beta, which is a result of inflammasome activation. But it also inhibits, PERK inhibitor also inhibits this in the wild-type cells. But if you have PERK and knockout, you don't see this reduction happening because there is no mitophagy going on. And you can also see IL-1 beta maturation here is not affected if there is no PERK. But in wild-type cells, you can see IL-1 maturation and caspase-1 active form are blocked by PERK inhibitor or ISRIB. So working out this pathway happens step by step. And so far, we have figured out that the observations we had from PERK inflammasome involved mitophagy. So this is what we figured. But it's not super clear how that happens because PERK and ATF4 activation, ATF4 is a transcription factor. So it wasn't immediately apparent how this transcription factor was really affecting mitochondrial clearance by autophagosomes. So we looked back to literature, and we noticed a clue from literature connecting ER stress to the mitochondria. So at the time we were doing these studies, ER's connection to mitochondria was not very well deciphered. So we looked for every clue we could find, and my student Özlem at the time recognized this study, which showed that tunicamycin, a general ER stress inducer, induced mitochondrial proteases such as long protease and YME1. The reason this caught us attention was that it was suggested that lone protease, even though it is in the mitochondrial matrix, might be able to cleave PINK1 kinase, which regulates PINK1 PERK independent mitophagy. So PINK1 kinase can potentially be threaded in enough to the mitochondrial matrix to be available for lone protease. So this is what the studies were suggesting. And biochemical studies also showed that interaction of PINK1 and lone P leads to cleavage of PINK1, that this would be a substrate for lone protease. So based on these studies, we explored whether PINK1 was a target of lone protease in our experimental conditions in macrophages, and whether PERK affected this dynamic. So here you can see that fatty acids like palmitate can induce lone protease in the mitochondrial matrix. And together with this, we see a dramatic reduction of PINK1 kinase, which drives mitochondrial clearance through mitophagy. However, if you inhibit the ISR using the approaches I told you about, the PERK kinase inhibitor or ISRIB, you see this regulation is blocked. So clearly, PERK's ability to regulate lone protease has a consequence on PINK1 in these cells. So the next thing we wanted to find out was, indeed, does lone protease affect PINK1 directly in these cells? So you can see that if we knock down lone protease, which is only a partial knockdown, it still has a huge effect on PINK1 accumulation in macrophages under ER stress conditions. So there's definitely a relationship between the protease and the substrate PINK1 in our experimental conditions. And so a little bit more evidence to the electron microscopy. I've shown you electron microscopy images of the induced mitophagy, but now I want to show you biochemical evidence from these cells. So you can see here that when we treat cells with palmitate right here, and together with the inhibition of the integrated stress response by the inhibitors or lone protease, we can see that on the mitochondrial fraction, so mitochondrial fractionated, we can see PERK and P62 adapter, which is very important for the mitophagy pathway, are recruited to the mitochondria. So when we inhibit the integrated stress response, we can see that mitophagy pathway assembles on the mitochondria. And moreover, if we look at a mitochondrial transporter, we can clearly see reduced numbers of the mitochondrial transporters, suggesting there's a reduction in mitochondria. Confirming this, we can see that the electron transport chain components, complex 1 to 5, are dramatically reduced if we treat the cells with the ISR inhibitors. So mitophagy is induced by biochemical measures as seen in the electron microscopy images, and this leads to a reduced number of mitochondria in these cells. So here we see, again, that lone protease, which is part of this pathway, has a consequence on the inflammation. You can see that if we knock down lone protease, palmitate does not induce the same inflammasome activation it used to. You can see reduction in caspase 1 and IL-1 beta, mature forms. And using a potent inhibitor of lone protease, which is very specific, you can see that inflammasome activation is ablated in macrophages. So I've really took you through this pathway. So it looks linear now, but it wasn't linear at first. So we can clearly see that lipids induce ER stress, which activates PERK, which leads to ATF4 translation. ATF4 transcription factor leads to increased amounts of lone P1, which is a mitochondrial protease, which uses PINK1 as a substrate under stress conditions. And this leads to this leads to a reduction in mitophagy. So when there is more damaged mitochondria because of the lipid stress and little mitophagy, we see accumulation of mitochondrial ROS, which is causally associated with NLRP inflammasome, and you saw with atherosclerosis. So what I tried to tell you is that our initial studies, which were published, shows that ISR inhibitors can prevent how lipids engaged inflammasome in macrophages, and this has dire consequences for atherosclerosis progression. And we are lucky at this stage because we have a wonderful molecule like ISRIB that targets this pathway without overt toxicity. And we know the mechanism, the molecular structure of this small molecule. And not only is it very, very effective in atherosclerosis, but current studies show that it has very important function in translational regulation in the brain. And it's been explored in traumatic brain injury and other diseases. So this is a very interesting molecule to follow forward with these studies. So I'm not going to have a lot of time to talk about this, but I wanted to tell you that this story didn't end here. So we're continuing because we made some really, really important observations. So obviously mitochondrial damage, mitophagy, the spill of mitochondrial ROS, mitochondrial DNA, those are really important things that the integrated stress response seems to control. But there is also a transcriptional element, an epigenetic regulation, if I may say, to how this immunometabolic regulation happens. And we couldn't blind ourselves to the fact that we saw that not only inflammasome activation was impacted in our studies, and we also saw that if we target the integrated stress response through the effector molecules like PERK, by inhibiting PERK kinase activity or by knocking down PERK, there was also a significant reduction in NLRP3 mRNA expression. And this was even more potent if we inhibited the integrated stress response core by ISRIB or knocking down the effector molecule ATF4. So these questions remained in our mind. And one of the very interesting things we noticed was that if we took the aortas from these studies and the macrophages, we looked at the DNA and we could see that 5-hydroxymethylation increased, whereas 5-methylcytosine decreased in these aortas. And also this is the case in the macrophages from these same studies. So this is very important because this is almost a readout for a TET epigenetic modifier, which is really important for atherosclerosis. So TET enzymes are very interesting. They have an important function in the nucleus in modifying methylated cytosine residues. So these are also very interesting because they also connect mitochondrial metabolism to the nucleus. So one of the things that regulates TET enzymes is the alpha-ketoglutarate produced from mitochondria, which is an important cofactor. And alpha-ketoglutarate can be directly converted to 2-hydroxyglutarate, which is an inhibitor. So this enzyme is a perfect candidate to study in our studies about how NLRP3 is regulated at the epigenetic level. So this is very important because studies have shown that TET2 is the second most commonly mutated gene in human clonal hematopoiesis. And TET2 hematopoietic cells have been shown to have activated NLRP3, more NLRP3, and more IL-1 beta induction. And TET2 deficiency in mouse models leads to atherosclerosis. So we are very interested in studying this connection from ER to mitochondria to TET enzymatic inactivation, not TET loss of function by genetic means, but TET loss of function by metabolic stress regulated through organelles. So I will leave you here in suspense because we're trying to finish up these studies, but I just want to mention that one great study led to another great study because of the imagination of these very young investigators who work in my lab. Most of them are PhD students, actually. So I want to thank Inci, who just graduated and doing a postdoc, Aslı, who did a lot of the mitochondria studies for their efforts. We couldn't have done this without really moving to the Cedars-Sinai Medical Center, because once we moved there, we landed next to an amazing mitochondria lab led by Roberta Gottlieb, and we learned so much about the mitochondria. So finally, ER and mitochondria synapse happened in our work. We could really delve into mitochondrial research with their help. We also are very, very thankful to Moshe Arditi and PK Shah because we got a lot of advice from them on how to connect our studies to the inflammasome activation and atherosclerosis. And the mice were created in collaboration with Rusty Lipward at Amgen. So there's a lot of funding agencies you see from Europe and the US. So our lab, connecting two continents, was funded by a lot of great agencies. So I'm really thankful for their support. And this is where I'm going to end and ask if you have any questions, please go ahead. Thank you so much to Davor and Dünya for hosting me. I could not hear you, Dünya.
Oh, yes. Dünya, you're muted. I'll just unmute you.
Oh, okay. Can you hear me now?
Yes, now we can.
Thank you so much for a fantastic talk and a beautiful work. If I may kick off, actually, the question and answer session with a couple of questions of my own, if you don't mind.
Do you mind if I just quickly use this time just to say that I will now run a poll. So feel free to just answer the poll whenever you like. You know, you can answer any time during the Q&A or even at the end of it. So just so that people know that. But yeah, please go ahead. That's all.
Yeah, it's here. Beautiful work. I have a question for you. Going back to the very beginning, when you described what triggers inflammasome and metabolic stress. Component of metabolic stress, obviously you've shown beautifully impact of lipids and fatty acids on activation of inflammasome in macrophages. But how about hyperglycemia? So what if you have, because macrophages are very metabolically active cells, so there's a constant crosstalk between demand for fatty acids and carbohydrates, similar to what happens in a heart. So have you completely excluded ability of hyperglycemia in these metabolic stress states to drive processes which you describe so elegantly relating to fatty acids and lipids?
So Dünya, thank you for that question. So even though I haven't studied hyperglycemia as a model. As you know, like hyperglycemia and hyperlipidemia coexist. So studying them together would be a great, I think, addition, and it would take this work further. I agree with you. So we haven't studied that, but we can't really separate those because,
yes.
Most people who come to the clinics are going to be suffering from both. You're absolutely right.
Exactly. And another quick one before I move on to, I'm sorry, is your ISRIB molecule, is it active, or is there an effect in other immune cells that are part of an inflammatory response, or is it macrophages specific?
So I'm the only one who studied this so far, so I'm really looking forward to labs to explore it in neutrophils, for example, T cells. We have absolutely no idea. The molecule was discovered several years ago, but immunology has not been interested in it yet.
Okay, thank you very much. I won't be selfish, and I will move over to a question and answer session because we have some very nice questions coming through. So if I read them for you, that'll be okay. So first question is from Professor Heinrich Tegmeyer. Fantastic, quick question. Why palmitate and not oleate?
Well, hi Heinrich. So palmitate stresses the cells, but oleate doesn't. So when we give palmitate or stearate or other saturated fatty acids versus their same carbon length fatty acids that are not saturated, oleate is monounsaturated, for example. They don't cause any stress on the cells. They don't cause an inflammatory response. They don't cause ER stress or mitochondrial stress. So it's been known for a long time that saturated fatty acids are the problem for the cells. And if you give them together, most of the cells that have a capacity to activate lipid storage pathways can try to package them and put them into lipid droplets with the help of oleate. So it's really the excess of the saturated fatty acids that's the problem.
Thank you. Next question from Gavin Mestay. Is LON constitutively active in the system?
So LON is constitutively expressed in the system. So in terms of its other substrates, what it does to them, I'm not sure, I don't know, but it's constitutively expressed. It's just being really induced by the stress response. That's all I know.
Thank you. Next question is from Chi Kung Lam. Great talk. What are other substrates for LOMP1?
Wonderful question. So it works in the mitochondrial matrix and it has a ton of substrates in the matrix. But I'm very, very curious to find out if LON protease produces, for example, other small peptides that would communicate these organelle stress responses. So that's an area that would involve a lot of skilled proteomics to understand if LON protease generates peptides that mitochondria spits out as interorganelle or intercellular communication. So some area that I couldn't really touch myself.
Thank you. Next question by Ron Magnozzi. Thank you for a fascinating talk. Have you seen any effects of inhibiting the ISR in macrophages on overall cytokine production or secretion other than inflammasome factors? That's the first part of the question.
Mm-hmm. So I have not looked thoroughly, like I haven't done a cytokine panel to see, or chemokine panel to see that. That would be interesting to look. We were really interested in the inflammasome because of our, selfish interest. We really love the inflammasome. But I don't know. And also the things that we looked at, TNF or other factors, MC1, they're highly regulated by the activation of the inflammasome in these cells. So we couldn't really dissect if those were direct or indirect.
The second part of the question is some ISR signaling needed for macrophages to deal with having to secrete many cytokines upon activation?
Probably there is a need because when you have a demand on ER production of proteins and folding, in fact, we do see, ISR components or UPR components are regulated during differentiation of immune cells, such as B cells into plasma cells. So, there is always basal activity of these pathways. They're homeostatic pathways, so they're not just a stress response. They become a stress response, but we know very little about their homeostatic regulation and their consequences for the cells. But these are pathways that need to be intact for a lot of functions.
Okay, next question is from Emiliano Medei, who says, great talk. Could other inflammasomes be involved in effect CAPS111 dependent?
They could
be effect CAP111 dependent.
So I would imagine inflammasomes that have the capacity to sense what is going on mitochondria would be clearly affected. For other ones, I can't imagine how.
Thank you. Emma Robinson, my mind is blown by the sophistication and your mechanistic data. Inspiring, thank you. In addition to having a role in white blood cells and atherosclerosis, NLRP3 also is well documented to play a role in ischemic heart disease. Have you looked in vivo models at her role of ISR inhibitors, also on heart function inflammation at baseline or in pathological models? P.S. In addition to TET, regulation and regulating NLRP3, GAD, and DNA damage response mediators are also affect DNA methylation.
Absolutely true. So there are some studies regarding the PERK pathway of the ISR in ischemic heart by, I believe, Glomboski's group in UC San Diego. So I don't do these studies myself, but I'm aware that there are some such studies. They haven't used ISRIB in those studies, for example, but it would be really interesting to see. And you're absolutely right. So even though, we were interested in TET activity because of the 5-hydroxymethylation, clearly activities that would change DNA methylation on cytosine residues would also be important. So we should definitely look even further. Yes, you're absolutely right.
Next question is by Emiliano Medei again. What about this mechanism on cardiomyocyte function?
Of the inflammasome, right? So macrophage, inflammasome, cardiomyocyte connection.
I presume so, because the question is just what about this mechanism on cardiomyocyte function? I'm not sure if it relates to Emma's question or his previous question, which was about CAPS111 dependency. I'm not sure.
Maybe he can clarify before we end.
Can you clarify what you meant by this mechanism? So Robert Chang Chicho asks, thanks for the amazing talk. Is a mitochondrial protein TSPO involved in mitophagy activity? Thanks.
I don't know the answer. We never looked, so so thank you for the suggestion.
Next question from anonymous attendee. Very interesting talk about a slide, the reduction in 5-MC. Do you anticipate global translation in these aortas to be less as a result of this reduction in 5-MC, or do you anticipate less 5-MC in other RNA classes, RNA or tRNAs?
So we don't really see, well, we haven't done, for example, a polysome profiling. So that would be an experiment to be done. But overall, looking at some other housekeeping proteins or genes, we don't see their levels completely changing, but it could be very specific to some areas of the chromosome. So that experiment needs to be done. And in terms of RNA classes, 5-MC, we haven't looked, but that would be an amazing area to go to. I agree.
Thank you. Jake Wen, wonderful talk. May we learn, how do we know ER is first damage induced by lipid stress, not mitochondria or nucleus?
It's really hard to know that because, as I mentioned, these are highly connected organelles. They co-synthesize things together, so they're not separated metabolically, and as you've seen, they're not separated signaling wise. So it's really hard to know which comes first, and I have given up on that question. Actually, I just see them getting stressed together in every place I look. So I'm focusing on fixing both of them rather than focusing on one. And signaling responses that connect them is a really important way to be able to fix them, because they communicate through these signaling responses too.
Thank you. Question from Matthias Marek. Beautiful data. Does palmitate activate the other branches of ER stress response beyond PERK?
Yes, it activates. So I haven't seen an inducer of the UPR that selectively induces one arm. So palmitate, like every other inducer, induces all three arms. But there are biological conditions in normal homeostatic conditions where we see, for example, during differentiation, one branch of the UPR being more prominent. So if it's not a stress response, we do see activation of one branch in physiological conditions.
Thank you. 3 more quick questions, because are you happy to continue answering? It's 6 o'clock, or we have 3 quick questions left. Is that okay?
Yeah, definitely.
Lovely. Lucy Carrier, is LUMP1 expressed in cardiomyocytes?
I believe it is ubiquitously expressed. I haven't, specifically looked at cardiomyocyte, but it should be expressed.
Emiliana Medei, did you observe it in other macrophages like cardiac resident macrophages?
Haven't looked.
Okay.
Thank you.
Last question from Venkatesh is, have you tried studying the effect of mitochondrially targeted antioxidants on the ROS inflammasome activation?
I haven't, but there's a lot of people who are studying that already on modulating the inflammasome response. Yeah, through targeting ROS. But my interest was understanding interorganelle communication, ER to mitochondria and mitochondria to nucleus, and how to modulate this interorganelle communication. So I didn't go to the ROS angle so much. Thank you.
Thank you very much. That's all 14 questions that you had after you talked with my cheeky 2 questions I asked at the beginning. So thank you very much for addressing all of them. I think that's all there is in terms of Q&A, Davor.
And, feel free to contact with me if you have questions later. My email is ebru.erbay@cshs.org. So,
Ebru, you can even write it in a chat if you like, so that might be easier.
Let me write it.
I wouldn't have been able to catch that, so.
Yes.
There is a chat button there, and you can just write it. I mean, in the meantime, I can probably just share the results of the poll. Thank you for people participating, but you can have a quick look through it whilst Ebru's filling in her email address.
Yeah, you can just take over. I'm just typing, so go ahead.
So sorry, I'll keep conducting these polls in the last few webinars just to kind of get an idea of what people are after. By the way, oh, there you go. It's Mike Shattuck saying in a Q&A, 101%. I'm not sure if that was the case. Perhaps it was. Yes, it was 101%. I'm not sure how that happened, Mike. Okay, well, on that note, I mean, first of all, I'd like to thank you personally. This is way beyond my comfort zone, so I'd like to thank Dunya for chairing. And Ebru, thank you for a really wonderful webinar with lots of questions and clearly lots of engagement from the attendees. We've still got 80 people here listening to us rattle on about the polls and percentages. So thank you once again.
Thank you.
Thank you, and see you all on Thursday.
Thank you so much. Have a great week.
Thank you. You too. Bye.
Bye.
Bye.